Hello, good morning, good afternoon, good evening, depending on where you're at. Welcome, everyone. I'm Matthew Heston. I manage the field bioinformaticians in North and South America. Thanks for joining us today for this exciting presentation. I'm joined here by my colleagues, Alex Sokel, who's our segment manager for cancer genomics, and Ki-Pen Kwa, who's our staff scientist in computational biology and bioinformatics. Um, Please do note that both of today's presentations have been pre-recorded, but actually the talks will be followed by a live questions and answers. So you're welcome to submit any questions you have during the talks by typing them in the area provided on your attendee control panel, and we'll get to those during the live Q&A. In addition, we've also uploaded two complimentary pieces of literature to your control panel, so feel free to download those and take those with you. So with that, let's get started with the first talk from Alex on an introduction to how HiFi reads can unlock cancer genomics.
Hi, everyone. Thanks so much for being here today. My name is Alex Sokel, and I am the segment lead for cancer genomics at PacBio. And I'm excited to tell you today about how our accurate long-read sequencing is already enabling the next generation of cancer genomics by providing comprehensive and multi-omic characterization of the cancer genome, epigenome, and transcriptome. So we all know that cancer is a disease of the genome, but much of the effects of the mutations are actually expressed at the level of the transcriptome and epigenome. This means that to really understand cancer, we need to be able to perform comprehensive and multi-omic profiling of these somatic changes. With accurate long reads from PacBio HiFi, you're able to see the complete cancer genome. So that's what we're showing on the left in blue, which includes not only small variants like SNVs and indels, but also structural variants, phasing, and direct detection of methylation. And then the complete cancer transcriptome on the right in magenta goes beyond gene expression to include detection of isoforms, fusions, and expressed mutations, which are then directly tied to things like neoantigen discovery. I'm going to start with the transcriptome since the rest of the talk will largely be focused on the genome. So I'm going to walk you through two quick examples where HiFi enabled detection of novel isoforms and fusions in both bulk and single cell RNA sequencing. So what do long reads enable in cancer transcriptomics? It's really the ability to characterize the full spectrum of RNA dysregulation in cancer. So most cancer transcriptome studies to date have looked solely at gene expression, whether this is by expression arrays, qPCR, or short reads RNA sequencing. However, we know there are a lot of other ways that RNA can be dysregulated in cancer, including differential isoform usage through alternative splicing, fusion transcripts resulting from structural variants in the genome, and somatic mutations that actually get expressed at the level of RNA. Estimates suggest that short reads can only reconstruct about 20 to 40% of the transcriptome. And so we expect that actually the importance of these other types of dysregulation have been underestimated thus far. So first, why are long reads so much better at reconstructing the transcriptome? On the left here in this colored diagram, you can see the exons of the gene in pink. And then below it, you see the multiple mRNA isoforms that are being produced by that gene. The challenge with short reads is that you can only identify the specific isoforms if you happen to have a read that spans a splice junction. So that's what we're showing with the dashed lines. And as I said on the last slide, we estimate that only about 20 to 40% of the transcriptome is able to be recovered in this way. In long read sequencing, on the other hand, which is what we're showing on the right, you just read off the entire transcript in one go. So you know exactly which isoforms each read came from without having to do any kind of assembly. And so because of this, again, we think that the role of differential isoform usage in cancer has been underestimated just because short reads do such a poor job of identifying them. And then one additional note here, in single cell RNA sequencing, this problem is actually even worse because typically in single cell assays, you're only sequencing the 3' end or sometimes the 5' end of the transcript. So then you really have zero ability to say anything about the isoforms that are present. So here's an example from a recent publication. On the left here, I'm showing a paper looking at isoform detection in HER2-positive breast cancer using bulk RNA sequencing. Here they found over 140,000 different splicing isoforms. And what was really incredible was that 67% of those isoforms were novel, meaning they'd never been seen before. That's what we're showing here in the aqua in the pie chart. And then when they looked further at these novel isoforms, they found that they were enriched in oncogenes. So actually they found 74 different isoforms in HER2, which was a huge number. And this is one of the best studied genes in cancer. And then they also found that many of these novel isoforms were missing conserved functional domains or had different predicted protein localization patterns, which indicates that these isoforms are likely performing a different biological function in these tumor cells. On the right, I'm showing a paper using single-cell RNA-seq in ovarian cancer to detect both isoforms and fusions. And so here I'm showing one of the novel fusions they detected with IGF2BP2 on the 5' end fused to TESPA1 on the 3' end. And because, as I mentioned, with single cell, you're usually only reading the 3' end of the transcript, this resulted in the fusion reads getting incorrectly labeled as overexpression of TESPA1 in the short read data, since TESPA1 was the fusion partner that's present on the 3' end. However, when we look at the same data in long reads, the long reads were able to correctly assign these reads to the novel fusion, and then you can see that the expression levels of TESPA1 are no longer elevated compared to normal cells. So I'll switch now to talk about complete genomes, which again include not only the small variants like SNVs and indels, but also SVs, phasing, and methylation. So here I'm going to first show an example from a semi-recent publication to give a sense of the scale of what remains to be discovered here. And then I'll show an example where we're able to detect both structural variants and small variants in the same phased haplotype, allowing us to reconstruct the somatic events that led to the development of the tumor. To give you a sense of the scale of what has been missed in cancer genomes using only short-read sequencing, this is a paper out of Mike Schatz's group from a few years ago, where they did long-read whole genome sequencing of the SKBR3 cell line, which is a really commonly used model for HER2-positive breast cancer. I've grown these cells in the lab. Many of you have probably grown these cells. But what was really impressive was when they took these commonly grown cells and performed long-read whole genome sequencing, they detected nearly 20,000 structural variants, and most of these had been completely missed by short-read sequencing of the same cell line. And then when they followed up to see whether these SVs were actually doing something biologically, they were able to confirm that a number of them did in fact result in fusion transcripts, indicating that they are indeed functional. So here we're looking at a region surrounding the P10 tumor suppressor gene in a different cell line. This is the COLO829 cell line. And I will mention that these data are found on our website in case you want to play around. But here what I'll show you is the ability to detect both small variants and structural variants in the same phased haplotype. So starting with the normal cells, what you can see, well, actually not sure if you can see this, but here upstream of the P10 locus, there is actually a heterozygous SNP. So half of the reads here have the reference allele and the other half have the alternate allele in lime green. That's in the normal. However, in the tumor at that same position, it actually looks like it's homozygous for the SNP, which we can infer to mean that two events occurred. First, there was a complete loss of the haplotype that contained the reference allele, and then a second smaller deletion in the remaining haplotype that's left occurred to span the P10 locus. So here we've lost both copies of this locus, resulting in a complete loss of function of the P10 gene. And the only reason we know which event happened on which haplotype is because the long reads give us the ability to phase these variants. And so again, if you want to play with these data and see what types of variation you can find for yourself, it's available to download at our website right there at the link. So the last thing that I want to note around cancer whole genomes is that with the release of the somatic Whittle analysis tool, which you will hear a great deal about more later from my colleague Ki Pin, we now have a complete sample to answer workflow for cancer whole genome sequencing. So you start with DNA extraction with our nanobind kits on the left to get high quality, high molecular weight DNA. And that then goes into our library prep kit, followed by sequencing on the Revio, and then analysis to perform variant calling for all of the types of DNA variants I mentioned, plus a lot more, which Ki Pin will dive into in just a minute. So all of this is to say that we're really excited about all of the things PacBio is doing to enable tomorrow's cancer genomics by offering complete solutions for long-read whole genome and transcriptome sequencing, because we think this will provide the comprehensive and multi-omic view of cancer that is really needed to understand it. Thanks, and I'll turn it over to Ki Pin.
Hi, everyone. Welcome to the webinar, and thank you for your time tuning into the webinar. Thank you, Alex, for the great introduction on what PacBio has to offer for cancer sequencing. In my session, I would like to walk you through the somatic widow workflow that we have developed for cancer genome analysis. As you can see, not only does PacBio offer the highest accuracy sequencing technology, we also strive to offer comprehensive analysis workflow to help you uncover new biology. I'm going to focus on the whole genome sequencing analysis workflow today, which is on the left. However, understanding cancer often requires not just the genome, but also the transcriptome. To that extent, we provide analysis tool to call fusion genes from iso-seq, our RNA sequencing solution, as well as a point-and-click interface to help you to discover novel isoforms in the heterogeneous cancer transcriptome. Those two tools are available both on GitHub for PB Fusion and on our graphical user interface software, SmartLink 13. As I mentioned, the focus today is on whole genome sequencing. We developed a widow workflow that consolidates our recommendations on how to analyze cancer whole genome sequencing data. By comprehensively benchmarking well-established and state-of-the-art tools on HiFi dataset, we were able to determine what are the tools that perform well on each of the aspects listed here. This includes small variance calling, copy number variance calling, structural variance, differentially methylated region, mutational signatures, reconstructions, and even homologous recombination deficiency prediction. To make everything easier for you, we collect the outputs from the workflow into an interactive report that anyone can open in their favorite web browser to quickly understand what's in the tumor. Now let's dive a little deeper on what the workflow does. Starting with HiFi reads for matched tumor and normal data, we align them to a reference genome with PBMM2. And we carry out depth and alignment quality assessments with MOSDEF and custom scripts. In parallel, small variants, structural variants, and copy number variants are called. Specifically for small variants, we offer two choices in Deep Somatic by Google or CLARIS by Hong Kong University. By using the small variants called, we attempt to phase the cancer genome in a diploid fashion, which will offer insights into which haplotype does any somatic variant originate from. We then annotate regions that are differentially methylated, comparing the tumor and the normal with a tool commonly used in short-read sequencing called DSS. For SV, structural variants, we use Cerberus to call and use a control panel from HBRC healthy cohort to reduce false positives. We also found purple to perform well for purity and ploidy inference, which is important for understanding tumor heterogeneity. The results from the full workflow are annotated, and we also provide a set of prioritized outputs based on entogen compendium of cancer genes. Here is an example of an interactive output for the Colo 829 dataset, which you can download from our website. The example report here is also available on the GitHub page for our somatic widow workflow if you would like to take a look at it. The report generated upon pipeline completion summarizes the depth and variant numbers, purity and ploidy that's inferred from purple, homologous recombination deficiency prediction using CORD, whole genome profile, whole genome copy number profile, small variance quality controls, mutational signatures, and a couple of tables that allows you to filter to the variants of interest. I would note that the criteria used for filtering and the reporting format here are really designed for a very quick first cut analysis. For any comprehensive in-depth analysis, you can find detailed outputs from the command line. Just letting the report run for a few seconds so you can take a look at what's available. Okay, I would like to now show you some of the benchmarking done behind the scene to ensure that our pipeline generates highly accurate results. Firstly, we demonstrated that HiFi cancer genome enables highly accurate variant calling, small or large. In both of the graphs, the blue curve represents 60-fold tumor coverage and 30-fold normal coverage, while the red curve represents 30-fold tumor coverage and 30-fold normal coverage. Both are reasonable experimental design for cancer whole genome sequencing. In structural variants, Severus is able to call more than 90% of the SVs validated in a 2022 paper for the Colo 829 cell line. When we manually inspected some of the missed SVs, we think they could be missed because of extremely low allele frequency, such as around 2%, or they could be specific to the cell culture that was sequenced in the paper due to the lack of reads evidence that we found in our data. It's important to note that even at 30-fold tumor coverage, we are still calling 86% of the true SVs, and that could actually be improved if we allow for a little bit more false positives by setting the reads threshold lower. We even did in silico titration and found that accuracy at 50% purity remains consistent, although sequencing deeper does indeed allow for better accuracy at these lower tumor purity settings. For small variants, we use the HCC1395 cell lines to benchmark the SNPs and indels performance. Using deep somatic, the plot shows performance at different allele frequency bins on the x-axis and F1 score of both the indel and SNPs on the y-axis. Single-nucleotide variants, SNV, are highly accurate with more than 99% F1 score at above 20% variant allele frequency. Indels are close to 90% F1 at 60-fold coverage, above 20% variant allele frequency. Even with deep somatic being new and being actively developed, the variant accuracy, calling accuracy is already very high, and the advantage of long reads in difficult genomic regions will certainly shine. To ensure that the small variants called are indeed accurate, we further looked into mutational signatures reconstructions from the HCC1395 cell lines, where there is an abundance of short-read sequencing dataset for us to compare to. Mutational signature is an extremely important aspect of cancer genome, where it elucidates the evolution history and mechanism of any tumor. If the small variants called are not accurate, we expect the signatures to also be inaccurate. Using single nucleotide variants from deep somatic for the HiFi dataset and STRUCA2 for the short reads dataset, we found that the mutational signatures reconstructed for both HiFi reads and short reads are basically identical, again reassuring that the variants called are highly accurate. On the slides, you can see the arrow pointing to the mutational signatures proportion that's contributed by different types of mutational signatures. And you can see that they are highly concordant between the Revio dataset and the short reads dataset. Another aspect of cancer genome that is very important is copy number variation. We know in germline dataset, HiFi reads are comparable to short reads in calling large copy number variants, but we also wanted to validate that in the tumor. We benchmarked two tools, Purple and CNV kit, on the Colo 829 dataset and compared them to the CNV segments that we extracted from a short read sequencing paper from 2022. As you can see on the screen, where the middle panel is the short reads copy number variants, and the top and bottom panels are the HiFi copy number variants using two different tools, Purple and CNV kit. You can see that all three different panels agree with each other on the copy number variants called across the entire chromosomes, which is on the x-axis. CNV is crucial for accurate clonality assessments, and it is therefore very important to validate that HiFi is able to recover copy number variations just as well as short-read dataset. Now that I've shown you the accuracy of HiFi reads, I would like to select a few examples of cancer variants that really highlight why we provide the best sequencing technology for cancer. This is an example that I think many of you may have seen before, but I always love to show these screenshots to everyone. What you are looking at is the readout of an important tumor suppressor gene, PTEN, in a cancer cell line. The top two tracks represent the diploid haplotypes from the matching normal cell line, whereas the bottom gray track shows the tumor haplotype, where you can see clearly there is a large 12 kilobase pair deletions on the right in the PTEN gene. On the left side, we color the CpG islands of PTEN promoters based on the methylation status. Red indicates methylated CpG sites, while blue represents unmethylated CpG sites. In addition to the large deletions on the right, you can see that the promoter region of PTEN on the left is clearly hypermethylated compared to the normal cell line, highlighting the complete picture HiFi sequencing can provide. Next, this is a collaboration between the University of Melbourne and us, where we attempt to understand a complex real cancer sample. This particular case was presented during the AACR annual meeting 2024. Previous efforts to characterize with short reads did not completely elucidate the oncogenic mechanism in this particular tumor sample. With HiFi reads, we can see that there is a tumor-specific hypermethylation of the regions corresponding to the BRCA2 promoter compared to the normal, which you can see in the middle. And this potentially shuts down the expression of BRCA2 from one of the two haplotypes. In the other haplotypes where there is no change in the methylation status, you can actually see there is a large 29 kilobase pair deletion completely resolved with HiFi reads. This is essentially a complete loss of function of the BRCA2 tumor suppressor genes by two completely separate mechanisms, one via structural variants and one via epigenetics, both of which we are able to directly visualize in the HiFi dataset without any separate sequencing effort. Another example of epigenetics in the cancer genome include methylation pattern in genes associated with epithelial mesenchymal transition. We know that HOXD9 is highly involved in this process, and using colo-A29 as an example, you can clearly see that there is a different methylation pattern comparing tumor on the top and normal on the bottom. You can imagine there are many such other genes in the cancer genomes just waiting for us to uncover their roles in the various oncogenic process. I hope I have now convinced you that PacBio provides one of the best technologies for cancer genomes, as well as a bioinformatics workflow that will allow you to discover new biology. Now, I would like to spend a few minutes on the boring part, which is how to run the workflow and what type of output does it provide. Besides the interactive report mentioned in the beginning, the workflow provides many useful outputs that you can use for in-depth analysis with your own scripts and codes. The workflow merely simplifies the steps and contains our recommended parameters to get the best results, because otherwise, there are many softwares that needs to be installed and optimized to work with HiFi reads. In PacBio, the majority of our command line workflows are written in the WDL language. WDL stands for Workflow Description Language. This is a workflow language that is highly flexible and supported by many popular workflow platforms, including Cromwell on Azure or AWS Health Omics. To run it offline on a HPC, high performance computing cluster, you can choose between Mini WDL and Cromwell. Both are well known to run WDL workflow. WDL workflow uses Singularity or Docker to handle software dependencies, and that's all you need when you want to run this workflow. You do, however, need to ensure that Singularity or Docker is available on your server. We test our workflow extensively on an internal Slurm cluster using Singularity, so that's the combo I'm most confident in that it will work smoothly. Having said that, we have customers running the workflow on other platforms such as Health Omics, Azure, or other HPC schedulers such as SGE. So those are definitely viable options too. The GitHub page provides a step-by-step workflow that you can follow along and run the Widow pipeline on our demo Colo 829 dataset. In the step-by-step workflow, the Colo 829 dataset is actually a small downsample dataset, but you can actually download a full dataset of HCC 1395 and Colo 829 that we use for benchmarking purpose from our official website, pacb.com. The input options to the workflow is in a JSON format, which is reasonably easy to understand, but I would describe them in a little bit more details here. The JSON consists of a few sections. The first one is a section that specifies where the BAM files are, and the second block specifies a set of reference files that you can download from Zenodo. Once you download the reference files, you place them in your server and you change the path on the screen to where the files are located on your server. In addition to that, resources for each step, such as memory as well as CPU, can be set separately for individual steps. We optimize the default of these resources to work well with between 64 to 128 CPUs, but if you have a smaller or larger servers, you can definitely be more generous or more stringent with the resource allocation. The last chunk at the bottom specifies path to a couple of large public databases that you can download. While they are optional, we highly recommend that you download them if you want to have the interactive analysis report. Without the annotations using this large public database, the pipeline will not generate a report. However, it will still run through all the steps and give you the raw VCFs if you want. Here are some example outputs you can find from the pipeline. Firstly, BAM files are phased with high phase or long phase, depending on what you choose. By default, this is high phase. Both normal and tumor BAMs are phased and output in the pipeline for you to visualize alignments if needed. We use MOSDEF to quantify the coverage in individual chromosomes as well as across all chromosomes. Alignment statistics such as percentage aligned, read length, and alignment concordance are summarized or even available on a per read basis should you decide to dive deeper into the statistics. On the right, you can see an example of MOSDEF tumor summary where it tells you that in chromosome 1, the mean coverage is 66-fold. On the second, third, and fourth chunk, you can see overall tumor alignment statistics, per alignment tumor statistics, as well as the align read length summary where it tells you what is the mean, median, and 50, and the total read length that's aligned to the reference genome. For SV, raw and filtered VCFs are available. Filtered refers to the VCF after excluding SVs that exist in the HBRC healthy controls. We also use a tool called Annotate SV to annotate the effect of SVs and the genes involved in the SVs. Lastly, the annotated file also goes through a prioritization process whereby only genes known to be implicated in cancers are kept. We use the Intogen Compendium of Cancer Genes for this prioritization purpose, as mentioned in the beginning. Looking at the right, you can see an example output from the annotated SV file that contains the gene names involved in a specific structural variants and the transcript length as well as the coding sequence length that overlaps with that particular structural variance. On the bottom is an example from the prioritized and annotated SVs, whereby you can find columns such as cancer type involved, rows of the genes, as well as whether there is a strong evidence that a specific gene drives the specific types of cancer. Similar to structural variants, small variants and methylation calls are also annotated and filtered. For single nucleotide variants, mutational signatures plots and raw data are both provided. For differentially methylated regions, the results are in TSV format, where you can find different categories such as promoter region, UTR region, large genes, as well as introns. The individual TSVs contain statistical significance of any groups of CpG islands that's differentially methylated comparing the tumor and the normal. Both the small variants as well as the differentially methylated regions are annotated similarly using Intogen to classify the genes involved according to their involvement in cancer. Last but not least, you can also find two types of copy number variants in the pipeline. One using purple and one with CNV kit, as we have compared in the previous slide. The difference between purple and CNV kit is that purple attempts to call subclonal copy number variants, which is in non-integer format, whereas CNV kit calls copy number in a clonal sense, where they are all in integers. Depending on the granularity of your research, both may be useful and can be used to cross-validate calls. On the right, you can see a circos plot generated by purple, where it visualizes across the different chromosomes what are the different copy numbers for any specific segments. You can also find tumor purity as well as ploidy from the output using from purple. That is all I want to talk about for the widow workflow. Thank you for your time again, and I will be happy to take any question.
Awesome. Thanks, Alex and Keepin. So yeah, we've had some questions come in. So let me start with a few that came in early on during Alex's part for her. First, with just a more general question, can you speak on the advantages of long reads versus short reads for cancer research?
For sure, yeah. I mean, I think the easiest way to sum it up is that long reads, accurate long reads from PacBio give you everything that you can get from short read sequencing in terms of small variants like SNVs and indels. But then really on top of that, with the long read information, you're able to get those additional types of variants. So structural variants, the phasing, and then also that direct methylation detection. So I think having those things on top of the small variants is really what sets the long read sequencing apart.
Awesome. Also, can you comment, you or Keepin, on the coverage requirements, and do we require a normal also?
Go for it, Keepin.
Yeah, sure. So we typically recommend to start with 30-fold coverage for both the tumor and normal. But in some cases, because tumor is highly heterogeneous, it can be tumor that has a lot of so-called low cellularity or purity, where you do need to sequence do a deeper coverage to uncover some of the variants that are of low allele frequency. So I think the short answer here is depends. So it depends on types of the tumor. Some tumors, blood cancer, for example, tends to have very, very low contamination. So you might be able to get away with 30-fold coverage. Some other tumors, you may want to actually go for 60-fold coverage. So that's the coverage that we currently recommend.
Awesome. I know this is a bioinformatics webinar, so let's just keep it generalized, but there was a question about DNA extractions. Can you get some general comments on those, Alex?
I can, yes. So really the main consideration when you're doing DNA extraction for long read sequencing is really to make sure that you have sufficient quantity of high molecular weight DNA. So I think I mentioned we have our own nanobind extraction kits, which are very good at this, but there's also a bunch of different kits that people have had success using as long as they result in high molecular weight DNA. So really you want to have a median fragment size above 30 kb on the fragment analyzer. So that's really the main consideration.
Yeah, wonderful. Keep in two general questions. One, I'll let you answer one each, and then I'll ask the next one. But one is, can you comment on the execution and runtime of the pipeline?
Yeah, so the execution runtime, typically on a 64 CPUs cluster or server, takes anywhere between 12 to 18 hours. So um, the the aspect that really affects the runtime is coverage. So that's the first one. So if you are sequencing 64 coverage, I'm seeing a runtime of anywhere between 16 to 18 hours. But for something that's 34, 12 hours, 14 hours, all that will take to finish the pipeline. Now, the other aspect that I want to mention is also, again, heterogeneity of tumor. I think you're seeing a pattern of how how I'm answering a lot of the cancer questions. It really depends on tumor type. If you have a tumor that, for example, has MSI, microsatellite instability, it tends to have a lot of variants that can kind of increase the runtime for example, the callers as well as some of the annotation steps. And that affects the memory usage as well, because when you have more variants that you need to annotate or analyze, then it takes more memory. So that's the two aspects that really affects the runtime. But I would say typically you can expect it to finish in less than 18 hours.
And then the second question, so you can definitely tell we've got some bioinformaticians on the line for this one, is what languages was the pipeline constructed in?
Yeah, so the pipeline is written in workflow description language, WDL. It's a very well-established language that's open source, so it has this GitHub page that describes the standard, the specifications, and it's a language that allows us to essentially define all the steps that we want to run. So, for example, you saw the diagram just now in the slides that talks about probably around 20 steps that you need to run to get the same results, right? So instead of asking you to install 20 softwares and type 20 commands, now you only have to at one single command and you get everything run in one shot. And of course, the parameters that's used for each of the softwares are tested and optimized to the best of our knowledge, of course, to make sure that you get a reasonable results by default when you run that one command. But all of the parameters are highly customizable. So if you have tumors that requires different parameters, you can always play with the parameters a little bit to see if you can get better results.
And then we had one that just came in that we'll repeat just for people to know, asking, does this require tumor normal pairs?
Yeah, good question. So that's probably the most frequently asked questions. Right now it does require a normal. We we will think about implementing a tumor only approach because I know it's highly sought after for some of the cancer types. The issue really is the mutation by tools. HiFi sequencing is still relatively new. There are a lot of the tools that's designed to call variants based on assumption that you have a normal, but when you don't have a normal, it's actually a very hard problem to tackle because it's hard to say whether a variant is somatic or germline when you don't have the germline samples to begin with. So I'm seeing a lot of some new tools that's popping up to do that. So we are also in the process of testing them, and again I will welcome anyone to reach out to me directly, for example, to discuss that. But just to put it shortly, right now the workflow does require a normal.
Thank you. Alex, a question for you. Great presentations on solutions for analyzing cancer genomes. As for analyzing cancer transcriptomes, can you comment on solutions for using HiFi reads to identify gene fusion events?
I can, yes. And thank you for asking that, actually. Yes, so this year we presented this whittle, this HiFi somatic whittle at AACR. Last year we presented a tool that we developed called PB Fusion, which does exactly that. So basically it takes the output of a long read transcriptome study and then calls fusions directly from it. So that's also on GitHub and is super well documented. So yeah, definitely would love for you to try that out.
Awesome. Keepin, I believe you mentioned it, but can you also answer, there's a question, are people already using this pipeline?
Yeah. Short answer, yes. So there are a couple of customers that I know that are already using the pipelines, and I think I mentioned in the talk that we test the workflow on a HPC that has learned, for example, but I've also heard of customers running it on the cloud. For example, so I've seen a couple of customers using it um, so far. I think the feedback has been good, but again, like I mentioned, tumors is highly heterogeneous. So I've had customers coming to me and say, "Hey, this results look a little bit weird." And after looking at it, it's usually because that tumor is of a specific type. It's a lot more complex than we imagined. So some of the parameters do have to be changed. Yeah.
Keepin, can you also comment on purple versus CNV kit? Any advantages or differences between those tools?
Yeah, so sure. So we have two output for copy number variants in the pipeline. So one is from CNV kit and one is from Purple. Now, the original reason for implementing Purple is that it estimates purity and quality of tumor, which is very crucial for looking at subclonality of variants, for example. So but what we have come to realize is there's a main difference in the output between CNV kit and Purple is that with CNV kit, it does a very simple segmentation of the read there. So it tells you that, okay, in this particular region, the copy number of is probably different from other regions, right? But it assigns a so-called integer copy number. So you say copy number is 2, copy number is 3, is 4, which is what you would expect from a germline sample, for example. But we know tumor tends to have, again, issue with heterogeneity. So you have a mix of different populations that can have different copy numbers. So imagine 50% of the cells have copy number of 3, 50% of the cells have copy number of 2, then the total copy number is not going to be either 3 or 2, it's going to be in the middle, right? So what Purple does is that it accounts for something like that, and it outputs a copy number that is non-integer. So you see something like 1.5, 2.5. So that kind of tells you, oh, okay, in this region, maybe there's two different populations of cells that's generating that different copy number. So that I would say is the main difference.
Thanks. In terms of how you evaluated the pipeline, can you comment on how you calculated the F1 score and also what you used as the ground truth specifically about structural variants?
Yeah, good question. So there are two aspects that we did benchmark on. One is on small variants, so SNPs and indels, and then the other one is on SB. For single nucleotide variants and small indels, we are using the HCC1395 cell lines. So we have the matched tumor normal cell line sequence, and this is a very, very well characterized cell lines where there is a set of variants that's curated based on, I think, sequencing on 60 different replicates. So on different instruments from different institutes on the same cell line, and then they curated a very, very high quality set of variants. So that's the set of variants that we compare against. So you can go and look for that set on the internet. So it's from SEQ-C2 consortium, SEQ-C2. Now, the other set of cell lines is actually the Colo 829. It's a melanoma cell line. So that's again another cell line that was previously characterized by multiple different platforms. So it was actually sequenced on PacBio, on ONT, on Bionano, a couple of platforms that really, as well as short read sequencing. and there was a set of SVs that's curated based on that cell lines in that paper, and then they actually did experimental validation, PCR, and a couple of other things. So a set of, I think, 60 over structural variants that has been validated. So again, we are using that set for the benchmarking purpose.
And maybe a bit related, you or Alex, can you comment on are any of these data sets that we've evaluated publicly available now?
They are. They're on our website. I don't think I can try to put the link in the chat actually, but if you search PacBio public data sets, you should be able to find it. Also on the GitHub itself, there are links to where you can download them on our website. It's probably the best documented GitHub I've ever seen, so definitely a lot of resources there. So if you want to download the data, yeah, we have links there.
And then there was also a general question, is there a step-by-step procedure or a document on how to analyze or use HiFi data? I'll first comment that, of course, it depends on the application. We have resources for lots of different applications, but being the subject of this talk, can you comment on documentation of using the somatic whittle?
Yeah. So so if you go to the somatic whittle GitHub page, you will find a step-by-step documentation. So basically, telling you where to download the data, the test data, how to actually install mini whittle, and all the commands that you need to essentially start to get the pipelines running, and some of the details on the output, and what are the options that you can change, for example, and small sections on frequently asked questions. So, for example, if you have a different schedulers, how do you get it running? So, yeah, there is a step-by-step documentation on the GitHub page. But if you follow it and then you can't get it running or anything, feel free to reach out to me. I'll make sure to help you to get it set up and running.
Yeah. And then you also had the great example where we had multiple mechanisms, including epigenetics. Can you comment on how to detect and analyze what we saw for the hypermethylation or hypomethylation?
Alex,
I can take that, yeah. In terms of our actual ability to detect the methylation, I can definitely explain how that works. It's one of my favorite things about HiFi data, actually. So the way that it works is for methylated Cs, it takes longer for the polymerase to incorporate the base as it's reading around the circular template. And so by looking at the incorporation of those fluorescent nucleotides over time and the kinetics traces, you can actually see whether or not it was a methylated C. And so what you get out as a result in the output from the Revio is actually what we call 5-base sequencing. So you have A, C, T, and G, obviously, and then also it will tell you all of the 5 MCs, whether or not those are methylated. And so, yeah, so that just is given to you directly as the data coming from the Revio.
Wonderful. With that, I think that's a good closing question, because I think we have to close up here. Couple closing remarks. Of course, first and foremost, thank you very much, Alex and Keep-In, for sharing your expertise and experience today. And also thank you everyone who joined for this webinar today. A few practical things. Also, in the next week or so, we'll send you an email with a link to today's recording, so you'll be able to go back, listen to any part of this again. Second, please keep up to date on upcoming PacBio webinars and events by visiting and bookmarking pacb.com/events. And finally, immediately following this webinar, you're going to receive a short questionnaire. Please do take the 2 minutes that is required to fill this out. It helps us understand your needs and also plan for future webinars. So thanks again for joining us today. I hope you'll join us again on a future webinar. And with that, take care and have a wonderful day. Thank you. Bye.
Thank you.
Yeah.
Bye.
Bye.